Hello and welcome everyone. In my last video, I gave an introduction about recombinant DNA and enzymes used for recombinant DNA and the vectors which are used. So, in this class, we will discuss further about the recombinant DNA technology. So, let's start with the host cells which are used in this recombinant technology. The ultimate aim of the development of recombinant DNA molecules or vectors is the multiplication of recombinant DNA by cloning in a suitable host cells. The kind of host cell which is used mainly depends on the aim of the cloning experiment. The host cell can be bacteria, yeast, plant, or animal cells. And the most commonly used host cell is E. coli. And the main advantages of E. coli is it is very simple, easy to handle. It grows rapidly and is able to accept and maintain a wide range of vectors. The doubling time of E. coli under ideal growth condition is 20 minutes. As the cell undergoes multiplication, the recombinant DNA within the cell also undergoes multiplication independent of its genome. And if the recombinant protein that we want to produce is of eukaryotic origin, it is better to opt for a eukaryotic host system such as yeast. And there are several advantages for yeast as an expression system. Yeast are the simplest of eukaryotic systems, and it is single-celled, genetically and physiologically well characterized, and it is very easy to grow and manipulate. Like bacteria, it can be cultivated in different volumes, either in flask, fermenters, or in industrial scales. There are several reasons for using a eukaryotic host system for the expression of a protein of eukaryotic origin. The polypeptide after synthesis, because the polypeptide after it is being synthesized has to undergo folding in the correct manner to attain its functionally active 3D structure. This requires the removal of some signal polypeptides or amino acids to initiate the process of folding to the correct three-dimensional structure essential for the production of functionally active protein. In addition to this, there may be removal or addition of or chemical modification of amino acids or the addition of other macromolecules such as carbohydrates or lipids to make the protein functionally active. All these processes together, which is called as post-translational modification of proteins. And the prokaryotic systems such as E. coli lack all these enzymes and other machinery essential for the post-translational modification of proteins. The eukaryotic protein induced in bacteria may fold incorrectly, resulting in the production of the functionally inactive molecule. Prokaryotic systems do not have the molecular machinery which is necessary for the splicing of introns of eukaryotic genes, resulting in the production of correct form of the mRNA. Hence, it is better to use the eukaryotic host system such as yeast as the host cells for the multiplication of the recombinant DNA. And the main process of the making the recombinant DNA is the first step is the construction of a recombinant DNA is isolation and purification of the vectors and the DNA fragments containing the gene to be cloned. There are a number of standardized procedures for isolation of plasmid and DNA fragments containing the gene. First digest the vector DNA with a suitable restriction enzymes and make it linear with or without sticky ends, which is determined by the type of restriction enzymes used, and then isolate the DNA fragment carrying the gene by digesting the genome or the source DNA strand with the same restriction enzyme used for the cutting the vector. And this digested vector and the target DNA cut with the same restriction enzymes are incubated together in the presence of DNA ligase. And during incubation, the sticky ends of the two DNA strands come together and by the base complementation and become hydrogen bonded to each other. At this point, a phosphodiesterase linkage is established between them, mediated by the DNA ligase enzyme. This results in the formation of recombinant DNA molecule of the vector and the target DNA, and this is the basic strategy for the construction of recombinant DNA molecule from a vector and target DNA fragment. The main disadvantage of this strategy is that there is a possibility of re-annealing reannealing of the sticky ends of the linearized vector, leading to the formation of vector itself without any foreign DNA fragment. This can be reduced by adopting any one of the following strategies, such as use of alkaline phosphatase, as I told earlier, or its linearization of the vector and the isolation of the foreign DNA fragment with two restriction enzymes instead of one, so that the two sticky ends of the vector will not be identical, which will prevent the re-annealing to some extent. And the next process is, after the recombinant DNA molecule is formed, the next process is the transgenics, which is also called as introduction of the recombinant DNA into the host cells. And this transgenics can be of 5 types. That is, the introduction can be done in by 5 major categories. And the first one is transformation. It is the most commonly adopted method for the introducing a recombinant DNA into a host cell. During this process of transformation, cells take up foreign DNA from their environment, but many cells, including E. coli and mammalian cells, are not able to take up the DNA from the environment naturally. Under such conditions, certain chemical treatments can enhance the ability of the cells to take up the foreign DNA or make the cell competent for transformation. This enhanced competency for transformation was found out by two scientists in 1970. They observed that E. coli cells become more competent to take up foreign DNA when these cells are incubated in cold calcium chloride solution. This mechanism is still followed for the transformation of E. coli cells as a part of gene cloning. And the next type of category, that is transfection. In this method, the recombinant DNA is mixed with the charged substances such as calcium phosphate, cationic liposomes, or any dextran molecules, and these are overlaid on the host cells. This ultimately results in the uptake of the external DNA by these host cells. And in the next method, that is electroporation, during this process, then electric current is used to create transient microscopic pores in the cell membrane of the host cells. Through these temporary openings, foreign DNA enters the cells. This method is especially suitable for cells such as yeast, mammalian cells, and plant protoplast. And the next one is microinjection. It is a specialized technique by which DNA fragment or gene can be directly injected into the nucleus of plant and animal cells. This method can be carried out without the use of any specialized vector. The procedure involves the direct injection of the DNA into the nucleus of host cells with the help of a glass microinjection tube or a syringe. The instrumentation for this process consists of a microscope of high magnification attached with fine needles, syringe, and other facilities to carry out the delivery of the DNA into the selected cells automatically. And the next method is biolistic method. It was developed for introducing foreign DNA into plant cells with the help of a gene or particle gun, which is also called as a gene gun. During this method, the microscopic particles of gold or tungsten coated with the DNA of interest is bombarded into the cells at high velocity. The bombardment of the particle is carried out with the help of the mechanical device called particle gun. Normally, the method of introducing foreign genes into the plant cell is done using Agrobacterium tumefaciens, but this bacterium is host specific and may not infect and transfer the gene in certain cases, particularly in the case of monocots. In the case of bacteriophage vectors, no special methods or treatments are required for inserting the DNA into the genome because the phage infection and the transfer of DNA into the genome is natural. Next one is identification of recombinants. And once the recombinant DNA is introduced into the host cell, it is followed by the selection of those cells which contain the recombinant vectors. During the process of genetic transformation, only a very low percentage of the total cell population takes the recombinant DNA. Therefore, it is very important to have an efficient screening method to select the transformed cells or those cells that have taken the foreign DNA. In most of the cases, there are many two stages of selection. First one is selection of transformed cells. The second one is to identify the transformed cells that have the recombinant plasmid. The transformed cells contain a plasmid and can be identified by the positive selection method. And the most common markers used for positive selection are antibiotic resistance gene present in the cloning vectors, or antibiotic resistance genes such as cell wall synthesis inhibitor or protein synthesis inhibitor such as ampicillins and tetracyclines. And the negative selection method can be adopted for identifying the presence of recombinant vector among the transformed cells in order to identify those plasmids carrying the foreign DNA fragment. The multiple cloning site in the vector is engineered in such a way that the insertion of a foreign DNA strand disrupts the expression of the selectable marker gene, and this phenomenon is known as insertional activation. And in order to use antibiotic resistance gene as well as the positive selection system, the plasmid vector must carry two different antibiotic resistance genes. And one such example of a vector which carries this both antibiotic resistance genes such as ampicillin and tetracycline are PBR322. And in a brief, this whole recombinant DNA technology is finished, and in brief, we will study the steps involved in recombinant DNA technology. The first step is selection and isolation of DNA insert, and which is to be cloned. The desired DNA segment is then isolated enzymatically. This DNA segment of interest is termed as DNA insert or foreign DNA or target DNA. And the second step is selection of suitable cloning vector. The cloning vector is a self-replicating DNA molecule in which the DNA insert is to be integrated. A suitable cloning vector is selected in the next step of recombinant DNA technology. Most common used vectors are plasmids and bacteriophages. Introduction of DNA insert into the vector to form recombinant DNA molecule. The target DNA, which has been extracted and cleaved enzymatically by the selective restriction enzymes, are now ligated by the enzyme ligase to vector DNA to form a recombinant DNA molecule, which is often called as cloning vector insert DNA construct. And this recombinant DNA molecule is introduced into a suitable host, and this suitable hosts are selected, and the recombinant DNA molecule so formed is introduced into the cells. And this process of introducing the recombinant DNA in the host cell is called transformation. Usually selected host cells are of bacteria from such as E. coli. However, yeast or fungi may also be utilized. And after this transformation, selection of transformed host cell is the next step. And in this step, the transformed cells are separated from the non-transformed cells by using various methods making use of marker genes and expression and multiplication of DNA insert in the host. Finally, it is to be ensured that the foreign DNA inserted into the vector DNA is expressing the desired character in the host cells. The transformed host cells are multiplied to obtain sufficient number of copies. If needed, such genes may also be transferred and expressed into another organisms. So this ends the basic principles of the recombinant DNA technology, and now we will study what are the applications of this recombinant DNA technology. And the first application is the production of transgenic plants. By utilizing the tools and techniques of genetic engineering, it is possible to produce transgenic plants or genetically modified plants. And many transgenic plants have been developed with better qualities like resistance to herbicides, insects, or viruses, or with expression of male sterility, etc. And another application is production of transgenic animals. By the use of recombinant DNA technology, desired genes can be inserted into the animal so as to produce the transgenic animal. The method of recombinant DNA technology aids the animal breeders to increase the speed and range of selective breeding in case of animals, and it also helps for the production of better farm animals so as to ensure commercial benefits. And another commercially important use of this transgenic animals is the production of certain proteins and pharmaceutical compounds. And transgenic animals also contribute for studying the gene functions in different animal species. And the biotechnologists have successfully produced transgenic sheep, pigs, rats, and cattle. And the next application of this recombinant DNA technology is the production of hormones. And bacterial cells such as E. coli are utilized for this production of different hormones or chemicals such as insulin, somatostatin, somatotropin, and P. endorphin. Human insulin hormone, or it is also known as humulin, is the first therapeutic product which was produced by the application of this recombinant DNA technology. And the next application is biosynthesis of interferons. And interferons are the glycoproteins which are produced in very minute amounts by the virus infected cells. Interferons have antiviral and even anti-cancerous properties. By this recombinant DNA technology, the gene of human fibroblast, which produces interference in the human beings, is inserted into the bacterial plasmid. These genetically engineered bacteria are cloned and cultured so that the gene is expressed and that interference are produced in fairly high quantities. This interference so produced is then extracted and purified. And this recombinant DNA technology is also used in the production of vaccines. These vaccines are the chemical preparations containing a pathogen which is weakened or which is in inactive state, and these vaccines may be given to human beings or animals to confer immunity to the infections. A number of vaccines have been synthesized biologically through this recombinant technology. These vaccines are effective against numerous diseases such as polio, malaria, cholera, hepatitis, rabies and smallpox, etc. The generation of DNA vaccines has revolutionized the approach of treatment of infectious disease. DNA vaccine is the preparation that contains the gene encoding an immunogenic protein from the concerned pathogen. And production of antibiotics. Antibiotics produced by microorganisms are very effective against different viral or bacterial diseases. Some important antibiotics are tetracycline, penicillin, streptomycin, and novacin, etc. Recombinant DNA technology helps in increasing the production of antibiotics by improving the microbial strains through modification of genetic characteristics. And it is also helpful in the production of some important chemicals, and few of them are alcohols and alcoholic beverages which are obtained through fermentation, and even organic acids like citric acid, acetic acid, and vitamins may be produced by microorganisms by use of this recombinant DNA technology. And application in the enzyme engineering, as we know that the enzymes are encoded by genes, so if there are any changes in the genes, then definitely the enzyme structure also changes. Enzyme engineering utilizes the same fact and can be explained as the modification of an enzyme structure by inducing alterations in the gene which encode for that particular enzyme. And the next application of this recombinant DNA technology is prevention and diagnosis of the disease. Genetically engineering methods and techniques have greatly solved the problem of conventional methods for diagnosis of diseases. It also provides methods for the prevention of number of diseases like AIDS, cholera, etc. Monoclonal antibodies are the useful tools for genetical disease diagnosis. Monoclonal antibiotics are produced by using the technique called hybridoma technology. And another example for application of this recombinant technology is gene therapy. Gene therapy is undoubtedly the most beneficial area of genetic engineering for human beings. It involves the delivery of the specific genes into human body to correct the disease. Thus, it is the treatment of disease by transfer and expression of the gene into the patient's cells so as to ensure the restoration of normal cellular activity. In application in forensic science. The application of recombinant DNA technology in forensic science have largely depend on the technique called DNA profiling or DNA fingerprinting. It enables us to identify any person by analyzing his hair roots, wood stain, and serum, etc. This DNA fingerprinting also helps to solve the problems of parentage and to identify the criminals by tracing the fingerprints or is any parts of the body such as hair, nails, etc. And it also aids in the biofuel production. And these biofuels are derived from the biomass, and these are renewable and cost effective. Genetic engineering plays an essential important role in beneficial and large scale production of biofuels such as biogas, biodiesel, etc. Genetic engineering helps to improve organisms for obtaining higher product yields and product tolerance. Practical applications of this genetic engineering recombinant DNA technology are: it is applied for localizing specific genes and sequencing of DNA or genes, study of the mechanism of gene regulation, molecular analysis of various diseases, and study of mutations in DNA, etc. So, this ends the recombinant DNA technology and its application.